Hi, my name is Dean Shaw. This is a lab report by me and Tina Hong. We tested the problem of how do factors affect the rate at which photosynthesis occurs. The specific factor we tested was the light intensity. Our hypothesis was the greater the light intensity, the more leaf chads will float due to the greater release of O2 from the higher rate of photosynthesis. The procedure consists of making leaf chads using a hole puncher, inserting these leaf chads into a syringe. Once the leaf chads are in a syringe, fill the syringe with sodium bicarbonate and create a vacuum to release all the air trapped within the leaves. To do this, put one finger over the top of the syringe and pull down with the plunger. What this does is creates a vacuum and allows the leaves to sink so we can determine when they float due to the release of oxygen. Once all the air has been released from the leaf chads, pour them into a cup full of sodium bicarbonate. Then move the cup into your desired location with the correct light intensity. Once moved to this location, set a timer for 15 minutes and check every minute to see how many leaf chads had floated up. Take data and then repeat for different steps. Two meaningful constants of the lab would be the same type of leaves to ensure that no variables were being changed, and also the CO2 concentration in the water, because that directly relates to the amount of oxygen being released.
In the lab with the light intensity variable, the group of leaf chads that had exposure to two lights had more leaf chads float up to the top than the leaf chads that had no exposure to direct light. By the end of the 15 minutes, all 10 leaf chads with more light exposure had floated to the top, while the leaf chads that had no exposure at all didn't float at all. The group of leaf chads with more light exposure had more leaf chads float because of the light and the amount that it was exposed to. The light was the reason why the leaf chads could float. The energy from the light was absorbed and used in photosynthesis, and because of photosynthesis, oxygen was released, and that helped the leaf chads float. The air bubbles of the oxygen will surround the leaf chads, making it able to float. The extra light also helped the leaf chads float to the top at a faster rate. Compared to the control group, the leaf chads that had exposure to light had all 10 leaf chads float by the 11th minute, while the control group only had 7 leaf chads float to the top by the end of 15 minutes. This shows how the rate of photosynthesis can be affected by how much light is present. The trial with no light had no leaf chads float. This means that no photosynthesis has occurred, and so no leaf chads could float. The trial with the two lights had all 10 leaf chads float. This shows that the light played a major role in making the leaf chads float. The leaf chads that had two lights floated at a faster rate than the control group. 50% of the leaf chads floated at the 13th minute in the control group, while the group with the two lights had 50% of the leaf chads float at the 9th minute. A source of error that was in this lab was that the light was heating the water/bicarbonate solution that the leaf chads were in, and so the temperature of each solution could have been different in each trial. An improvement to that would be to cover up the cup of the solution with a clear covering, like, for example, another cup with water, so that the heat would hit the water and the covering, but the light will still be absorbed by the leaf chads. Another source of error is the lighting of the room that the trial was conducted in. The lighting of the room could have possibly affected how fast photosynthesis could occur, and it could have helped leaf chads float faster. An improvement to this would be to do the lab trial in a dark room so that no additional lighting would be present and only the lights that are being part of the independent variable would be present. This would make sure that only the lights that are being tested could affect how the leaf chads float. An extension to this lab could be to use different kinds of lights, for example, using LEDs against fluorescent lighting. 2 LEDs could be used to see how many leaf chads float in 15 minutes, and 2 fluorescent lights could also be used to see how many leaf chads float in 15 minutes. LED lights would be expected to have more leaf chads float at a faster rate because their wavelength is more narrow and shorter than fluorescent lighting's wavelength, and that means that LED lightings will have more energy than fluorescent lighting.
Thank you all so much for watching. Have a very Merry Christmas. Dean and Tina, signing off.